BM754, and I'll be presenting my research PanCan diagnosed, in which I developed a series of algorithms for the accurate and affordable early diagnosis of pancreatic cancer via machine learning and bioinformatics. Pancreatic cancer is the fourth largest cause of cancer death, as 85% of people die. This happens for two reasons: a lack of early diagnosis and misdiagnosis. As shown in Figure 1, the pancreas is found behind the stomach, making imaging unclear and early diagnosis difficult. Thus, no screening scans for pancreatic cancer exist, making the 5-year survival rate stay at a constant 10%, as shown in Graph 2. NCBI links this to the flaws in diagnosis, stating that survival rate will increase to 40% if the majority of cases are diagnosed early. Another problem in pancreatic cancer is that it is underdiagnosed as diabetes 20.1% of the time because of its similarity in symptoms and gene expression. Current machine learning research achieves low accuracy, around 70 to 75%, because the algorithms don't account for the early diagnosis and misdiagnosis while being expensive. This therefore led me to my goal of developing an early diagnostic tool for pancreatic cancer by analyzing the expression of 19 genes. The success of the algorithms was credited to 4 different characteristics, which included knowing the difference between the 4 groups here, as well as early and late stages at an accuracy of over 90%, providing a user interface to aid in availability, and supplying an affordable diagnosis. This led me to my 3-phase project, the first being the development of 5 algorithms, which included the logistic regression model, a KNN, a random forest, a naive Bayes, and a neural network. In Python's Jupyter Notebook, 408 samples of all 4 groups were inputted from the sources shown here, and was split into no diabetes and diabetes data. The samples with no diabetes were then split into an 80% training dataset and 20% validation dataset. Training data was then fit onto one of the algorithms, and the validation data obtained the best classifier from the list defined below 1,000 times. The most frequent classifier was then used to retain the following scores of all diabetes patients. The whole algorithm was then iterated 500 times, and all values were averaged. This was then repeated for all of the algorithms. In the second phase, a user interface called PanCan diagnosis was made. Here, users could choose what algorithm they wanted, the patient number, and the total iteration. This was run, and the patient number, algorithm used, and accuracy was displayed. One can see this as patient number 105 had pancreatic cancer with 90% accuracy using a KNN. As shown in Graph 5, the first part of the engineering goal was achieved for all except the random forest, and the second part was achieved for all except the random forest and logistic regression models. Therefore, the ideal algorithms for this research were the KNN, naive Bayes, and neural network, as they had high performance rates in every aspect of the project. Nevertheless, these algorithms can diagnose pancreatic cancer at exceptionally high rates of 95%, 20% more accurately than previous research. An additional application from this project is the user interface, which allows for an affordable system when diagnosing pancreatic cancer, cheaper than the MRI scans that exist today. Microarrays also allow for the detection of gene expression, making the user interface accessible. In the third phase, the reasoning behind the algorithm's high performance was evaluated by investigating the connection of 19 genes, as shown in Figure 3. Using StringDB, it was found that these genes were involved in the cytoplasm red, cytoskeleton blue, and nucleus green at confidence intervals of over 0.9, as shown in Graph 7. Bioinformatics tools like PathCards also help find expression levels of these genes in the pancreas, as shown in Graph 8. A microarray, which consisted of S00, S01, S10, and S11 groups at different pancreatic cancer stages, were additionally analyzed using ImageJ. As shown in Graph 6, when pancreatic cancer progressed, there was a decrease in the area of the cytoplasm, a significant increase in the area of the cell, the area of the nucleus, and aspect ratios of all cellular components. However, when diabetes was present, the change was more dramatic, showing how the expression of the 19 genes differed in the presence of diabetes and pancreatic cancer. However, this project had some limitations, including the small sample size and inaccuracies in machine learning. To improve this project, future modifications like instructing the algorithms to tell the difference between early and late stage samples and solidifying the expression of the genes through in situ hybridization could be done. In the end, PanCan user interface and bioinformatics provides a mechanism to diagnose pancreatic cancer in its early stages accurately and affordably. Thank you.